Welcome to students. Today we are going to discuss the second part of our plant growth and development. In this session we are going to discuss about differentiation, de-differentiation and re-differentiation. These three important sectors that is related to growth of plant will be discussed in this session. So you see, differentiation is a process in which the meristem cells, it may be root apical and shoot apical meristem, and cambium differentiate and mature to perform specific functions. Here a new term is there with my you, that is cambium. It is specialized tissue, though it is considered as a permanent tissue, but it can regain its cell division ability. That's why it is essential for the secondary growth usually. So in this, cell walls and protoplasm undergo structural changes. Capacity of cell division is lost. Once the meristematic cell is converted to permanent tissue, the cell division capacity is already lost. That is also seen in the cambium, but sometimes during the growth period, it again regain the division ability. That is the specialty of the cambium. So, in example, if you see loss of protoplasm to form a tracheary element, they also develop very strong, elastic, lignocellulosic secondary cell walls. That means it contains both the materials, that is lignin as well as cellulose. So secondary cell wall actually makes the cells more stronger and tough to carry water to long distance even under extreme tensions. That means these cells, those who are permanently changed themselves into tracheary elements, they cannot revert back to their normal conditions and they are unable to continue the cell division process. So this is called differentiation. That means a normal cell that is converted to a specialized cell and which may lose the capacity of cell divisions. Now you see, under certain conditions, living differentiated cells regain the capacity of division. Just before few moments I told you about cambium, and that is the specialized cells. Though they are living cells, they have not died yet. So they regain the cell division process, and this is called de-differentiation. That means once differentiation is made, and it is also again getting back to its original status of cell division. So in example, if we are looking at the cambium, formation of meristems in cambium and cork cambium from differentiated parenchyma cells. So we are finding then here you may look at this. You see the stem secondary growth, which is explained in this diagram in the left side. It is magnified. The green part in the diagram representing the de-differentiated cells, those who are performing the cell division and contributing towards the secondary growth. So similarly, next you see, de-differentiated cells can produce cells that again lose the capacity to divide but mature to perform specific functions, and it is called re-differentiation. That means these cells which are once de-differentiated, they can produce some new cells which are again getting their permanent situation, and re-differentiation occurs and those cells are performing the specific functions. So plant growth is open. It can be indeterminate or determinate. Both kind of growth can be seen. So differentiation is also open because cells of tissues arising out of the same meristem have different structures at maturity. So though the cells are coming from the same ancestral meristematic cells, on maturity they show some differences because of their specificity in their job. So final structure at maturity of cell, tissue is also determined by the location of the cell. So in example, cells positions away from the root apical meristem differentiate as root cap cells, while those pushed to periphery mature as epidermis. So here you can see the differences between the cells. Those are produced from the same meristems. So after this, let's see some more points on plant development. So it is the process that includes all changes in the life cycle of an organism, from seed germination to senescence. So it is sum of growth and differentiation. It is represented as follows. You see in this flow chart, meristematic cells through their cell division and differentiation, they produce mature cells. Mature cells may also de-differentiate, and gradually these mature cells comes under a phase that is called senescence and gradually turn to death. This is the life cycle of a plant cell. The period just before the death is known as senescence, where the physiological activities, the chemical reactions are usually reduced and gradually the cell is moving towards the death. Now, plants follow different pathways in response to environment or phases of life to form different kinds of structures. This ability is called plasticity. So, according to the environmental response, plant the phases of life to form different kinds of structures. This is called plasticity. Here in diagram you can see juvenile leaf, terrestrial habitat, adult, and in water habitat. How the plants, same plants, their leaf structures are somehow different in different environments. Heterophily, due to phases of life, example in cotton, coriander, and larkspur, the leaves of the juvenile plant and mature plants are different in shape. And heterophily due to environment, example, difference in shape of leaves produced in air and water. If the plant is living in air, then its structure somehow differs, right? The plants living in water also showing a difference. That means environmental change may cause the change in their development. Similarly, there are different factors controlling plant development. That are intrinsic factors and extrinsic factors. Intrinsic factors, they are coming under, they are present inside the cells, like intracellular factors, that is genetic, or intracellular plant growth regulators. They might be hormones. And what are the extrinsic factors? They are light, temperature, water, oxygen, and nutrition. Next section, we are dealing with the plant hormone. That will be uploaded on Thursday because I will be absent in coming 2 days. Thank you.